Hi, good afternoon, everyone. I know it's around 12 and people are probably still going to be logging in, but for the sake of time, we're going to go ahead and get started. Thank you all for attending our GHUCCTS KL2 Visiting Scholars Seminar. The KL2 Visiting Scholar Program was actually intended to be a in-person series, seminar series, and it's a collaboration between Virginia Commonwealth University, VCU, and the GHUCCTS program here. And we were, unfortunate that COVID hit when it did, but fortunate that Zoom can actually allow us to reach a much broader audience. So thank you all for coming today. And we hope that this is going to be a very informative meeting for everyone that's attending. We hope that this form of the Visiting Scholar program will become a model for KL2 programs nationally. So thank you very much for your participation in that. My name is Annie Kruger. I am an assistant professor in the Department of Gastroenterology, and I'm also a KL2 scholar. I'm here with my fellow KL2 scholar, Dr. Emeka Ihemalandu, who's an associate professor in the Department of Surgical Oncology. And we're both very pleased to welcome our keynote speaker and fellow KL2 scholar, Dr. Julian Zhu, today from VCU. So Julian will be giving us a glimpse into some of his cutting-edge research, and his talk is entitled Albumin Hitchhiking Delivery of Versatile Drugs to Tumor and Lymph Nodes for Cancer Immunotherapy and Chemotherapy. To tell you a little bit more about Julian, he received his PhD from the University of Florida in 2013 and did postdocs at the Cancer Research Center and the NIH. He is now an assistant professor of pharmaceutics and pharmaceutical engineering and sciences at VCU, and he's a prolific scientist. What's apparent from his body of literature is that Julian's generated, and he just started really doing research about a decade ago. He's already generated over 80 peer-reviewed papers, of which 23 are either first author or senior author in journals such as Nature Communications, Science Advances, and PNAS. And what's become apparent is that Julian is a very arduous scientist who is dedicated to developing clinically relevant cancer therapies. His research goal is to develop novel drug delivery systems to efficiently target cancer cells in various tissues of the body. So please welcome me in joining Julian to the GHUCCTS seminar series. Just please note that we will hold any questions you may have to the end of Julian's talk, when Dr. Ihemalandu and I will moderate the Q&A session. So please go ahead and use your Q&A Zoom tab to ask your questions, and we will compile them at the end of the talk and ask Dr. Zhu about those questions, okay? And without any further ado, please allow me to introduce Dr. Julian Zhu.
All right, thank you. Thank you, Annie. Can you all hear me all right?
Yes. Okay.
My name is Julian. I'm assistant professor from Virginia Commonwealth University, fellow KL2 scholar. And today it's my pleasure to discuss with you about some of my previous and current research in my postdoc area and in my current research group. We're going to discuss the drug delivery. More often, we're going to discuss immune drug delivery, often to lymph node, sometimes also chemo drug delivery to tumor. But the whole purpose is to potentiate tumor immunotherapy. Because we're going to mostly discuss immunotherapy, I need to show this background in which Dr. Chen, Daniel Chen, A.R. Merman summarized a very interesting cancer immunity cycle. There are multiple nodes in which that can be modulated for immunotherapy, and we're specifically interested in this node, central in this node for cancer antigen presentation. Specifically, we were interested to deliver subunit antigen and vaccine and adjuvant together for immunotherapy. For antigen, we're often interested in tumor-associated or very recently tumor neoantigen, which are exclusively in tumor cells, not in healthy cells, because of their generated by somatic mutation or other genetic events. And we also work a lot on adjuvant. So here are some examples of traditional CpG oligonucleotide, A4A small molecule. And recently, STING agonists are some of our interests as well. And very often, those are used in combination with chemotherapy or other immune checkpoint blockade combination, or even radiotherapy. So this is one of our favorite organ or tissue, lymph nodes. Lymph node is basically a ball of immune cells. This area will have a B cell follicle. In the center, we have T cells, and afferent lymph and lymphatic vessel will bring a lot of antigen presenting cell, so that these antigen presenting cells can interact with B cell and T cell and elicit adaptive immune responses. And then those adaptive immune cells or cytokines can be secreted to blood circulation for tumor immunotherapy. And then to the cellular level, antigen presenting cells, the antigen can be processed if needed, and then complex with MHC, and this whole complex can be transported on the cell surface for antigen presentation, and that eventually for tumor therapy can elicit T cell responses, CD4 or CD8 T cell responses. Those tissues are important for vaccine delivery because at the tissue level and at the cellular level, we need to have an approach that can deliver vaccine molecules into the lymph node tissue and inside antigen presenting cells. And however, molecular vaccine, especially for subunit peptide vaccine, very often they're small. When they're locally injected, they're disseminated randomly and resulting in a poor accumulation in lymph node tissue, but sometimes even have toxicity because of their accumulation, their dissemination into the blood. If we have a carrier such as a nano carrier, in the form of a nano vaccine can be drained more efficiently through lymphatic vessel into lymph nodes. That will result in better chance for them to interact with immune cells in the lymph nodes, so that will have a better immune response and lower toxicity. So today I'm going to talk about mainly two types of drug delivery system that can do this. We also work on micromaterial that could be used for this type of or other type of application, but we're not going to discuss this today. So first, I'm going to discuss with you about albumin hitchhiking drug delivery system that can be used to deliver subunit vaccine to lymph nodes, as well as chemotherapy, small molecule chemotherapy drug, and nucleic acid oligo drugs systematically. This line of research started from my postdoc work with Dr. Sean Chen at NIBIB NIH. So Dr. Sean Chen's lab developed a derivative for Evans blue. This Evans blue can bind to albumin avidly, and this derivative can also use this metamer functionalized group to conjugate with any biomolecule, say imaging agent or therapeutic agent, as long as there's a reactive group. The conjugate can have a decent binding affinity with albumin, 1 micromolar. When injected a radiolabeled compound into an lymphedema patient in which the lymph drainage was blocked. Of course, you can appreciate well the lymph drainage was blocked, but also you can see the lymph nodes were lighted up by this compound. So we thought this is an interesting approach if we wanted to deliver anything to lymph nodes, and what needs to be delivered to lymph node, and vaccine comes up with as an example. So that's why we start to use this molecule to deliver vaccine to lymph nodes. And also, if we IV inject this compound to a healthy volunteer, and hours after injection, you can still appreciate high intensity in blood circulation or in blood-rich organs. also some in bladder. So this can be used for systemic delivery of drugs, say chemo or some oligo drugs. Actually, lymph node accumulation by some dyes, for example, evans blue, has been observed in clinic for many years and has been used to isolate lymph nodes in sentinel lymph node analysis, and because they can bind to endogenous proteins such as albumin. And interestingly, albumin has many receptors. One of the receptors is neonatal FC receptor FcRn, which is highly expressed in a couple of antigen-presenting cells, which are pretty interesting to us because we wanted to deliver vaccine molecule to antigen-presenting cells. So that's what then we respectively conjugate this melamine evans blue with an adjuvant called CpG oligonucleotide, and conjugate another monomer evans blue with a peptide antigen, and we mix them. Then we call this mixture a albumin-binding vax, AlbiVax. When we inject this AlbiVax locally, subcutaneously into the body, they will form complex with albumin, and this complex behave like a nanoparticle in efficiently drain and retain in the lymph nodes. In the lymph node, they can also interact with antigen-presenting cell, deliver vaccine molecule into antigen-presenting cell, elicit T cell responses for cancer immunotherapy. And very often, this will also upregulate the expression of some immune checkpoint molecules. And then if we combine vaccine with immune checkpoint blockade, then that can lead to an even better therapeutic response. So this is an imaging study in which we wanted to really quantify how much vaccine molecule can be delivered into lymph nodes. So we label this compound with a radioisotope copper-64 through this nodal chelator, and we have a couple of different derivatives of this molecule in which we have either we have a pack or we have no pack for those groups, and we have different lengths of linker between MEB and the CpG. And now we regulate all of them, and then we subcutaneously inject those compounds at the tail base, so that this compound can drain bilaterally through lymphatic vessels to inguinal lymph node and to axillary lymph node. And at different time point after injection, we can do PET imaging to quantify the amount of a vaccine molecule in a specific tissue, such as the lymph node. And shown here is an example. 6 hours after injection of this compound, you can appreciate the accumulation in the inguinal as well as the axillary lymph nodes. We can do that for all these compounds, and you can see that all those albumin-binding candidates, no matter how long the linker is, they can have a appreciably higher accumulation in the lymph nodes compared with other controls. Of specific interest, not only conjugate CpG has no accumulation in lymph node, but also a commonly used enzyme oil and water emulsion has no accumulation in the lymph node other than trapping the vaccine in the injection depot. And also a pegylated polyethylene glycol molecule, which has a very close molecular size as the albumin, but even though this conjugate has very poor accumulation in the lymph node, which tells us that not only the size, but also perhaps the biology of albumin helps with the delivery and retention into in the lymph nodes. If we quantify the accumulation of a different compound in the lymph node, then we can tell that if we have 2 copies of linker, the accumulation seems to be the highest. Then this is the compound we end up using in the rest of the study. And we can also quantify that in the rest of in other major tissue and organs, and we confirm that in the lymph nodes there are a lot of accumulation. You can also note that in liver, in kidney, there quite some accumulation as well. Though this is a personality per gram, means the density of the compound. But if you calculate the mass, liver and kidney also has a lot. So this can also be used for liver and kidney delivery as well. We wanted to look further and see at the tissue and cellular level how those vaccine molecules were distributed. So in the first study, we label this compound, the vaccine compound, with a fluorophore dye, and we inject the vaccine same way into mice. 1 day after injection, we isolate the lymph nodes. We clear the lymph nodes so that we can see through the lymph node tissue for fluorescence imaging. And if we use a fluorescence imaging, tissue imaging, we can actually tell the very abundant distribution of vaccine molecule inside the lymph node, especially in subcapsular cells. And if we zoom in, we can see some molecules start to migrate inside into the B cell or even T cell follicles. At cellular level, we image this albumin-binding CpG using a fluorophore labeled CpG, and we can appreciate in this whole single cell, in those each dot is a endolysosome. We can appreciate that the red signal on the surface, on the membrane of the endolysosome, probably indicate the compound are bound to that receptor, toll-like receptor 9, which are distributed on the endolysosome membrane. I'm not going to play the video, but you also confirm that again, this is one cell. If we label fluorescence, we label the vaccine and mouse serum albumin respectively, we can see that by their co-localization of the fluorescence signal, that they're uptaken together into the cell and the lysosome. And because the co-delivery of adjuvant and antigen into each antigen-presenting cell is very important for vaccines, for subunit vaccine. So we look at the adjuvant CpG labeled with 1 fluorophore, and a model albumin-binding antigen with another fluorophore. We incubate them together with antigen-presenting cell, bone marrow-derived dendritic cells, and after a time, a period of incubation, we then use confocal microscopy and look at the co-localization. And again, this is one cell. We can appreciate that a lot of co-localized fluorescence from adjuvant and antigen, which is great for their optimal immune modulation and therapeutic efficacy. We sort of recapitulate the previous two co-delivered study in vivo, and we saw that especially in dendritic cell and macrophage, a lot of albumin and CpG adjuvant or co-delivered, especially on the early 1 day. And for adjuvant and antigen, no matter on day 1 and day 3 after injection, there are a lot of co-delivery in dendritic cell and the macrophage. Not so much in B cell, not as much in B cell compared to DC and macrophage in this case. Another interesting observation is that if we wanted to look at the duration of the antigen presentation. So what we did in this study is we label a spherical model antigen with a fluorophore FITC, and we compare this unconjugated free vaccine with this albumin binder conjugated AlbiVax, and we incubate them with a DC dendritic cell. After some time, after 8 hours, we wash the cells so that anything outside cells will be washed away. And for those free vaccine, after we wash, then the signal started to fade away. And then for the AlbiVax group, if we wash them, the signal keeps strong for up to 2 days until we stop the experiment. So this is a very interesting, sustainable antigen presentation. We think this is because that the efficient uptake of this AlbiVax created a reservoir of vaccine inside cells so that it can keep supplying vaccine molecule for antigen presentation. And also, because we also care that the safety, we actually look at the AlbiVax compared with a free CpG. It decreased the toxicity. One looked at by the reduced splenary magnet, in which the spleen was enlarged, and second by the acute systemic inflammation, in which the blood cytokine, such as L6 and L2, was induced in a less acute manner, which is good to prevent some acute inflammatory toxicity. We can do that for peptide antigen as well. One type of antigen is neoantigen, which are only generated in tumor cells but not in healthy tissue because they are generated by somatic mutation. So they are very, very personalized, very selective. And this is some example of neoantigen discovery. We used a neoantigen called ADPGK discovery reported in 2014. We confirmed the presence of this neoepitope in the cell line, MC38 murine colorectal cancer we used. And we conjugated the peptide with evans blue. We can also conjugate other peptide, but the ADPGK was shown as an example. We did the same PET imaging quantification, and we confirmed that the albumin-binding formulation can dramatically increase the accumulation of peptide antigen in the lymph nodes. We then tried, for example, for this melanoma model in a B16F10 immune-competent mice. We inoculate a B16F10 tumor, and we used the AlbiVax, in which includes albumin-binding CpG adjuvant and the albumin-binding TRP2 TRP2 antigen. And this mixture already helped increase the therapeutic efficacy compared with the free, as you can see, the red curve, compared with a free vaccine, or compared with the vaccine that was emulsified by a common adjuvant IFA, showing the orange curve. But this is not good enough. We then combine the vaccine, AlbiVax, with the antibody that block PD-1. So that combination helped with the immunotherapy, further slow down the tumor growth. But none of the tumor were regressed. We thought maybe chemotherapy can help, and we choose the one Abraxane, one chemotherapy drug that was tested in clinic for immunotherapy, and we saw that it can further decrease the tumor growth. But once we stop chemotherapy, then the tumor start to grow back. So there's a lot more work to be done, currently undergoing in the lab to further increase the therapeutic efficacy for some of the very immunosuppressive tumors, such as this model. We sort of confirmed this study in a non-metastatic melanoma model, in which the melanoma tumor deposited in the lung tissue and then treated in the similar way as previously, and we saw a similar trend of the therapeutic efficacy. Another further extension of the application is to use the vaccine to elicit immune response so that it can help to promote a phenomenon called the abscopal effect. So what happens in abscopal effect is the radiation, such as ionizable radiation, can of a local tumor can help cure the rest of a distant tumor. The underlying mechanism is by immune activation, but the frequency is very rare in clinic. if we can have a way to increase the chance of abscopal effect, then it's going to be very significant. The radiation we use is photodynamic therapy, and we use radio and radio photosensitizer. We use a photo and a photosensitizer called HPPH. We load this photosensitizer molecule into a nanoparticle. And then in a mouse model in which one mouse has two tumors on the left and right side, and then we inject a HPPH photosensitizer nanoparticle, and then we only irradiate the left tumor, but not the right tumor. And after irradiation by laser, we then inject vaccine for immune stimulation. And then the vaccine will train to elicit immune response. We observed that in the photodynamic therapy treated tumor, of course, the combination is much better than either of the monotherapy, but also in the non-PDT treated tumor, in the index tumor, we also observed a similar efficacy. So this tells that the immune activation, systemic immune activation, help to treat not only the directly irradiated, but also the index tumor, which was not directly irradiated. So we then also tested immunotherapy, immune modulation for neoantigen. So this is a neoantigen that we just showed for ADPGK, and we, after injection for twice and 2 weeks apart, and on week 3, we analyzed the blood T cells, and we found that this AlbiVax indeed increased the percentage of antigen-presenting, antigen-specific CD8 T cells. And this is accompanied by a high immune memory phenomenon. And also especially in the antigen-specific CD8 T cells, there's an upregulation of PD-1, and that's why we eventually use anti-PD-1 for combination. And in this model, similar to the previous B16F10, when we treat with the vaccine itself in this model actually behaved not too bad, even though the complete regression rate was pretty low after 2 injections, 2 treatments. And a combination of the anti-PD-1 really increased the therapeutic efficacy, especially the rate of complete regression. In this case, eventually 6 out of 10 tumors were completely regressed without recurrent within 3 months when we stopped the experiment. Okay, this is pretty much about the Evans Blue albumin-binding based vaccine delivery. I wanted to switch the gear a little bit and talk about a little bit about this approach for systemic chemotherapy drug delivery. As you can remember, you can appreciate a high level of Evans Blue molecule signal in the blood or blood-rich organs along hours after injection, IV injection. So we then conjugate this compound with chemotherapy drug. One of the chemo drug is MMAE, a very potent drug that itself cannot be used as a drug because it's too toxic, but it was used in a couple of FDA-approved antibody drug conjugates. And we bought this commercially available bulky molecule that has a functional group that can be used by us to conjugate with albumin-binding molecule. And after this compound is taken up into the cells, this linkage, VC linker, can be cleaved by cathepsin B, which is highly expressed in a lot of cancer cells. And after that, this part can also be self-immortalized to generate an intact MMAE molecule. And we care a lot about the pharmacokinetics. And so we measure the drug delivery distribution by quantitative PET imaging. And in this albumin-binding version and in this free drug, in this tumor model, we observe that the tumor uptake is much higher than in the albumin-binding version than in the free drug version. And consistently, blood retention is also higher in this group. The half-life, the blood circulation half-life was increased by 10 times for the albumin-binding version compared with a free version. And this is very helpful. This was translated to a better therapeutic efficacy in this model. And one injection of 3 mg per kilo, typical dose, cured 4 out of 5 mice tumors, and eventually only 1 tumor grows back. So this is a very interesting preliminary data that we hope to push further. Not only chemo, small molecule chemo drug, some oligonucleotides such as a DNA, RNA, aptamer, or even siRNA can have very short half-life. So this is an aptamer, RNA aptamer, that's about 30 oligonucleotides that target EGFR. As you can appreciate that this aptamer can bind specifically to the different level of EGFR in 3 different tumor models, but the uptake is pretty low. So if we conjugate aptamer with an albumin-binding molecule in the blue, then the uptake is much higher, as you can differentiate from by the 20 times of difference of scale bar. If we quantify them for the free aptamer, we only have this much in the tumor, and it is cleared very rapidly, as you can tell. Only at 1.5 hours after injection, we only have very little. And then if we have an albumin-binding version, then the difference is very striking. So we think this can be a useful tool to deliver oligo or anything with a short half-life, but needs a longer circulation half-life or tissue uptake. So next, I'm gonna talk a little bit about polymer nanoparticles, which we also study a lot for small molecule delivery, especially also study a lot about nucleic acid delivery. So we started this work when I was a PhD student at University of Florida. We developed a method to synthesize a type of a nucleic acid nanoparticle. I'm not going to dive into the details, but eventually we can get a nucleic acid nanoparticle, and then we can load we can choose different nucleic acid sequence just by designing different template for the synthesis. And in this case, when we synthesize the CpG adjuvant into the nanoparticle, we can directly inject this microparticle, as you can tell by the size, into tumor. And that microparticle of CpG itself can already slow down the tumor growth of this hard to treat B16F10 melanoma tumor. But this is not enough. When we stop the treatment, it grows back rapidly. So we thought 2 ways can help this. First, to increase the potency of the adjuvant. The one way is to increase to add more adjuvant. One adjuvant is an shRNA that can silence a immunosuppressive STAT3 in antigen-presenting cells. The other is by shrinking the shrink the particle from microparticle to smaller nanoparticle, so that their drainage to lymph node when we inject them subcutaneously is more efficiently. And second, we can load hydrophobic neoantigen peptide into the particle, so that the neoantigen can help to elicit tumor-specific immune responses. Those are the data that confirm the size shrinkage as well as the morphology. And this indeed helped with the immune response. First, it's specific to antigen that we used, and second, it's more potent. In this case, we used the MC38 tumor that's specific to the ADPCK antigen. And we can do similar. We can use a similar principle, but a simpler approach in which we use the micelle, a polymer micelle, which has a hydrophobic core in the center. And this hydrophobic core can be used to load hydrophobic small molecule immune adjuvants, such as a RA48, A2-like receptor 7 and 8 agonist. And on the shell, we can conjugate with DNA, and this DNA can be used to synthesize long CpG adjuvant. So now we have 2 adjuvant, and by doing so, we also have a simple but a bi-adjuvant nanoparticle nanovaccine. And this we show that this is better than mono adjuvant, and in an immune study, we show that this can potentiate the tumor-specific T-cell responses. I'm not going to discuss much about the details, but the principle is that if we use nanoparticle to co-deliver multiple adjuvant together with adjuvant, together with a peptide antigen, then the tumor-specific immune modulation can be further promoted. So then I'll talk a little bit about 2 types of research in which, 2 areas in which we wanted to modulate immune responses by targeting the nucleic acid sensing pathway. One is the DNA sensing, called CGAS-C. The other is RNA sensing, called ADAR. I will talk about one example about the DNA sensing. So this pathway, CGAS-STING pathway, is a natural immune defense pathway used by our own cells. So in this pathway, if we have a type of a molecule called a cyclic dinucleotide, such as cyclic GA monophosphate or cyclic GMP, which we're going to discuss in the following example, then those molecules can activate STING, and then the STING activation can eventually trigger the production of type 1 interferon responses, and that can be used for cancer immunotherapy. And also, even though we're not going to discuss much about today, if we have a long double-strand DNA in the cytosol, which is, as you know, not normal, then this can trigger the activation of CGAS. And CGAS can use our own ATP and GTP as a substrate to synthesize the CGAP. And now the CGAP can, again, trigger this signaling pathway. But today I'm going to discuss how an example in which we use the nanoparticle to deliver this GMP to go through this pathway. As you can tell from the structure, it's basically a very small hydrophobic nucleus sensitive molecule. It's not easy to deliver. Not even easy to synthesize a nanoparticle to load this molecule. So we developed a particle in which we utilized a polymer nanoparticle as a base, and on the surface of a nanoparticle, we used a DNA that can interact with the CGMP. The reason it can interact because they have a lot of C in the DNA, repetitive C. So now this C and G can form sort of a hybridization, CG hybridization. And interestingly, this repetitive C sequence, they can transform their conformation at a lower pH to a tetramer called an emotive C quadruplex. Now this formation will lead to the release of CGMP. Now this CGG is kicked out and released from the nanoparticle. And this is what we envision will happen inside an endolysosome, which is acidic after they are taken up into the immune cells. And then we use this to study tumor immunotherapy and modulate the immune microenvironment, tumor immune microenvironment. This is the conjugation. This is to confirm the conjugation of the DNA and the polymer by either by the absorption or by GL electrophoresis. And this is the size, about 180 nanometer, 100 nanometer. There's a potential because of the negative charged DNA as well as the CDG. Those particles are negative charged. The control of liposomal, cationic liposomal nanoparticle is positively charged, but it was used as a control. And we confirmed by MST, microscale thermophoresis, we confirmed the binding of the CDG with a C-rich repetitive C motif DNA. The binding affinity was about 47 nanomolar, which is very distant. And interestingly, because the nanoparticle can prevent nucleus from accessing the CDG, it can actually protect the CDG from degradation. As you can tell from this study, in which we incubate this compound, two formulations in cell culture medium, the free CDG was degraded much faster than the STING nanoparticle. And also in different buffer solution, which have a different pH condition, it showed that the CDG release was pH responsive. This means that at a lower pH, the response can be very efficient. Within half an hour, half of them were released, and within 1 hour, over 60% were released, even though later it will plateau in our condition. But the pH responsive CDG release is going to be important for their release in the endolysosome. And this particle can be efficiently internalized into dendritic cells and the macrophage, even more efficiently than a liposome, a very commonly used nanoparticle liposome formulation, especially at the later time points. And under confocal, if we stain the endolysosome with an tracker and label the CDG with a fluorophore, and so we can observe their distribution inside cells, we can observe that not only they can be efficiently taken up by the immune cells, but also if we quantify the fluorescence of a CDG inside and outside the endolysosome, and then we calculate the ratio, signal ratio of outside over inside endolysosome, we found that the outside over inside fluorescence intensity ratio was much higher for STING nanovaccine than liposome nanovaccine, which means that this nanoparticle can facilitate the endosome escape of CDG, which is important for CDG to access and interact and activate the receptor STING in the cytosol. And after that, we used a model cell line that was treated to behave like a M2 pro-tumor macrophage, M2-like macrophage. And after we treat them with our STING nanovaccine shown in the green circle, as well as the rest of the control, in which we either have an untreated macrophage or M2 macrophage, or 5C nanoparticle, which means the only nanoparticle, because we'll have a 5 repetitive C, or free CDG, or fluoride modified CDG, which was used to stabilize CDG from nucleus degradation. Lipo-CDG is a liposome cationic liposome CDG, and M1 is a positive control. And we showed that by either the cytokine secretion or by the marker, co-stimulatory marker, or MHC2 expression, we showed that the STING vaccine can upregulate the pro-inflammatory factor expression, or those co-stimulatory factor expression. That means that the M2 was converted in a way to M1 anti-tumor type of macrophage. And this was confirmed by RT-PCR result for of those markers. And not only in mouse murine cells, but also in human THP1 monocytes, the STING nanovaccine can actually activate the immune responses very efficiently, pretty much comparable with a lipo CDG, which is important for their future clinical study. And in a tumor, B16F10 melanoma tumor, if we inject 5 times of this nanovaccine into tumor directly, and we can tell by RT-PCR that the tumor microenvironment was the immunosuppression was decreased in the tumor microenvironment by the nanovaccine, and this was translated to a better therapeutic efficacy in which the tumor growth was slowed down, the survival was better, but we have to stop this study long earlier than we expected because of practical issues. But we're hoping to resume this study and study how we can further combine this with rationally with other immune checkpoints or other potential synergistic therapeutics. So I think this is about it. I hope I have introduced you 2 types of drug delivery system based on albumin, based on polymer nanoparticles. I wanted to note that we're very interested to develop potentially clinically translatable nano drug delivery system using very trustable, reliable materials, such as those event glue derivative or those very commonly used lipid nanoparticle or polymer nanoparticle materials. So in the end, I'd like to thank many of our group members who did all the hard work. Of course, my postdoc advisor, Dr. Shan Chen, under which I did a lot of albumin-binding study, and support from a lot of internal as well as NCATS KL2 program, as well as and support from my Center for Pharmaceutical Engineering and Sciences, School of Pharmacy. And that's it. Thank you. Hope you're well.
Thank you very much, Julian. So that was an eye-opening presentation. So we have a question for you here, and I'll just read out the questions as they come, and you can answer, and as they come, keep coming. So the first question says, what steps will it take to move from these model systems to human trials? Are there specific practical or regulatory hurdles along the way?
Yeah, this is a question that we're very interested in, and we keep in mind always. There are a lot of drug delivery systems, even a lot of nanoparticles. The reason we actually tend more and more to well-established or more and more simple drug delivery systems, such as this albumin-binding molecule and very well-established polymer or lipid nanoparticle are potentially easy CMC, easy clinical translation. As you can tell, for example, for the albumin-binding drug delivery system, even though eventually the drug delivery behave like a nanoparticle, but it's actually a small molecule. And that small molecule was derived from a event glue that was used in clinic for a century. And we had some preliminary study that the safety in not only mice or monkey, but also in human are pretty promising. So those are the factors that we consider when we decide which which drug delivery system to use. And back to that question, we think this can help us to interact with FDA for clinical translation. This can facilitate our conversation, facilitate our study in clinic.
Thank you. Any other questions?
I have another question, Julian. So you showed that some of the albumin hitchhike molecules do accumulate in the kidney and the liver. And I was just curious, when you then attach either a drug or like a chemotherapy to this, if it accumulates more in the kidney or liver, could that lead to greater kidney and liver toxicity potentially? Or is it, do you do you foresee that it should actually offset some of the toxicity? That's one question. The second question is, some of the, for example, PD-1 antibodies for melanoma have resulted in toxicities that create autoimmune conditions in patients, like they'll create colitis or they'll create autoimmune hepatitis. And being a gastroenterologist, I see a lot of this in my practice. So I'm just curious how your drug delivery system could either benefit or potentially worsen, I don't know, or have you thought about what it might do to some of those side effects of these checkpoint inhibitor drugs? Two questions.
Thank you. Good question. Yeah, great. Thank you. Question. First question, toxicity. This is really a case-by-case situation. What I mean is specifically for, depends on what drug or even what conjugation, what potential toxicity they may have. Definitely, we very often, no matter how we inject the albumin-binding compound or many of the nanoparticles, we often see a lot in liver. And for some compounds, some formulation, we also see a lot in other tissues like lung or kidney. So whether it is bad, it is toxic for that tissue, we have to really study and see. But this is definitely something that we need to evaluate, we need to test. But the good thing about that is this can also be an opportunity if you wanted to deliver anything to liver or say to lung or a kidney, then that can also be an opportunity. But if we wanted to deliver say the vaccine to lymph nodes, but we saw a lot in the liver and kidney, then we have to evaluate that. In our case, we have not, because of the vaccine itself is not like a chemo drug and not like a very toxic chemo drug. We did not see much toxicity in that case. And second question, I think that's a issue of specificity. If we can elicit tumor-specific immune response, then the chance of autoimmune response can be elicited to some extent, can be lowered down. Immune checkpoint blockades, they are specific to the immune checkpoint, but they're not, the immune checkpoint is not 100% specific to tumor. So I think that's a issue. That's basically why there is some non-specific autoimmune response. If we can have a vaccine that can elicit tumor-specific immune responses, and even that combined with the immune checkpoint blockade, and because now we have that specificity, and now we have stronger immune responses to tumor, but relatively less to non-tumor cells.
All right, great. Julian, I've got 3 questions for you here. The first 2 are kind of similar, but I'll just read out to you. So it says, you've shown us 3 cool complementary approaches, each with great examples. How do you decide how to best match delivery modes with specific drug, vaccines, or target diseases? And then the second question is a follow-up to what Annie had been asking you. What's the most promising example of a nanoparticle therapeutics? So,
appreciate. Well, I think that, or again, depends on what drug or what indication that we wanted to address. Say, for example, if for the albumin-binding drug delivery system, we have a few requirements. For example, this drug has to be able to be conjugated. Chemistry-wise, we'll have to touch it a little bit without affecting, at least without disrupting the therapeutic efficacy. And there are many drugs that cannot do that, honestly, even though we try hard, but chemistry-wise, it's just not as easy as many other drugs. And for those drugs, maybe those physically loading nanoparticles, polymer or even liposome lipid nanoparticle can be a better choice. And the second is, let's see, for example, for lipid nanoparticles, they are unique because they can have charges, and then we often use it to deliver oligos or deliver large nucleic acid, say DNA, RNA. In those cases, it's not easy or not even practical to conjugate those molecules with an albumin, small molecule albumin binder, and then efficiently deliver to the target tissue or cells. And then in those cases, a complexation, more coarse complexation could be more practical. I think to the second question is also related, depends on what specific application, specific drug that we're going to use. See, for example, there are already a couple of nano formulation approved by FDA. CBG, for example, approved in a liposomal formulation as a vaccine for hepatitis B, I think, prevention. And the SRA, in recent years, a couple of SRA was also approved for liver complications. They were delivered by one by a lipid nanoparticle, one by a small molecule conjugate that target GalNAc in the liver. And also there are a lot of mRNA initiative. You may have heard that COVID-19 vaccine study done by Moderna and NIBIB. They were using ionizable lipid nanoparticle to deliver large mRNA molecule. So I think, again, it depends on the specific drug and the specific condition, complication, disease indication that we're going to address.
All right, great. So we got just about 2 minutes, and I have one last question there. So the question is asking, with accumulation in the liver or kidney, could you revisit the cell targeting to macrophages or autophagocytic cells versus targeting to cells with specific markers or transporters?
Great question. We have all the targeting drug delivery system I've discussed today are more or less in a passive fashion, and so there's a room that we can increase the targeting or delivery specificity. That is simply by using a targeting ligand. I asked you about the LGR5 earlier today. The purpose is this: if we can have a peptide or a ligand that can target a specific receptor for that disease indication, then the delivery can be more specific. See, we may have even higher accumulation in tumor, but lower accumulation in non-tumor tissues or cells. So those are the future effort that we wanted to invest.
All right, we're out of time. It's 12:59. I wanted to thank you, Julian. This has been actually very wonderful. It's been great having you on here on campus. And we look forward to collaborating with you. I'm sure there are a lot of people who have heard your talk who want to be reaching out to you.
Thank you.
I want to also, again, congratulate you on the great work you've done. It's actually quite impressive and, to say the least, a bit intimidating for some of us. But that's a good thing. We need more people like you, to push forward.
Thank you.
Want to thank you, and wish you all the best as you continue to forge, this path, cutting edge.
Thank you. Thank you.
Thank you very much.
Wish the same to you. Thank you all.